Hello and welcome, medical students. Today, we will delve into the fascinating world of porphyrinogens and their intermediates, the heme synthesis pathway, and the various aspects surrounding heme metabolism. By the end of this lecture, you will have gained a comprehensive understanding of the intricacies of heme synthesis, its regulation, and its vital role in various physiological and pathological processes. To comprehend heme synthesis, it is imperative to first understand the importance of porphyrinogens and their intermediates. Porphyrinogens are the precursors of heme, and these molecules play a crucial role in the formation of the heme structure. The synthesis of porphyrinogens occurs primarily in the mitochondria, with various enzymatic reactions catalyzing the stepwise conversion of aminolevulinic acid, ALA, to porphyrinogens. Moving swiftly along our journey of heme synthesis, we now explore the intermediates involved in this intricate metabolic pathway. The heme synthesis pathway consists of several crucial intermediates, including porphobilinogen, PBG, oroporphyrinogen 3, ura-IE, coproporphyrinogen 3, copra-IE, and protoporphyrinogen 9, protox. Each of these intermediates undergoes distinct enzymatic reactions, resulting in the sequential buildup of the heme molecule. While porphyrinogens and heme play essential roles in biological processes, they also possess the potential for harm. Porphyrin molecules have a unique ability to absorb light energy, which can result in the generation of reactive oxygen species, ROS, through a process known as porphyrin phototoxicity. Excessive ROS production can lead to cellular damage and tissue injury, particularly in skin and liver tissues. This phenomenon has important implications for patients with certain types of porphyrias. A deficiency in heme synthesis can have far-reaching consequences for human health. Disorders such as porphyrias, characterized by defects in specific enzymes involved in heme synthesis, can lead to a buildup of toxic intermediates and heme deficiency. This imbalance in cellular heme homeostasis can result in a myriad of symptoms, including neurological manifestations, photosensitivity, and even life-threatening crises. Accurate diagnosis and proper management of these conditions are crucial to mitigate the potential complications that arise from heme deficiency. Heme levels within cells need to be tightly regulated to ensure optimal metabolic function. Intriguingly, cells possess intricate heme sensing mechanisms that allow them to adjust heme synthesis and degradation in response to fluctuating heme levels. Key players in heme sensing and regulation include heme-regulated eukaryotic initiation factor 2 alpha kinase, HRIK, heme-responsive transcription factors, and heme-responsive element binding protein, HERP. Together, these molecular actors orchestrate a delicate balance to maintain cellular homeostasis. Diving deeper into the world of heme metabolism, we explore the intricate regulation of heme biosynthetic enzymes. Each step of the heme synthesis pathway is regulated by a specific enzyme, and alterations in these enzymes can have significant implications for heme production. The regulation of heme biosynthetic enzymes occurs at both transcriptional and post-translational levels, ensuring fine-tuned control over heme synthesis. Heme oxygenase, HO, enzymes play a pivotal role in heme catabolism, resulting in the release of biliverdin, iron, and carbon monoxide. There are two key isoforms of heme oxygenase, HO1 and HO2. HO1 is an inducible enzyme primarily activated in response to oxidative stress and inflammation, while HO2 is constitutively expressed and involved in maintaining cellular heme homeostasis. Understanding the functional differences between these two isoforms provides essential insights into the broader biological roles of heme metabolism. Heme metabolism and oxidative stress are intimately connected. On the one hand, heme can act as a pro-oxidant, generating ROS and contributing to oxidative damage. On the other hand, oxidative stress can impact heme synthesis and degradation, potentially disrupting cellular heme homeostasis. The delicate balance between heme and oxidative stress highlights the intricate interplay between these processes and their impact on cellular physiology. Beyond its role in oxygen transport, heme has emerged as a key modulator of the immune system. Heme can influence immune cell functions, such as macrophage polarization, T-cell activation, and cytokine production. Additionally, heme can activate immune pattern recognition receptors, leading to the production of inflammatory mediators. Understanding the complex interactions between heme and immune regulation offers exciting prospects for the development of novel therapeutic strategies targeting immune-related disorders. In recent years, our understanding of heme has expanded beyond its classical roles to encompass signaling functions. Heme can serve as a signaling molecule, participating in various intracellular signaling pathways, including those involved in cell proliferation, apoptosis, and differentiation. The versatility of heme as a signaling molecule underscores its importance in diverse cellular processes and highlights its potential as a therapeutic target. Unraveling the multifaceted relationship between heme and cancer is an area of active research. Heme metabolism, biosynthetic enzymes, and signaling pathways have been implicated in both cancer promotion and suppression. Hemoproteins, such as cytochromes, also play critical roles in redox reactions and maintain normal cellular functions. An in-depth understanding of heme's involvement in cancer biology may pave the way for novel diagnostic and therapeutic approaches in oncology. The management of porphyrias presents a complex challenge due to the diverse spectrum of these rare disorders. Treatment strategies primarily focus on symptom management, preventing acute crises, and minimizing phototoxicity. Therapeutic options range from lifestyle modifications, such as sun protection and avoidance of triggering factors, to pharmacological interventions targeting specific steps of the heme biosynthetic pathway. A multidisciplinary approach involving dermatologists, geneticists, and other specialists is crucial to provide optimal care for individuals with porphyrias. In conclusion, the intricate world of heme metabolism and its associated pathways hold immense significance in both health and disease. Our exploration today has shed light on the importance of porphyrinogens, intermediates, heme sensing and regulation, heme oxygenase isoforms, and the complex interplay between heme, oxidative stress, immunity, and cancer. Understanding these intricacies is vital for the development of targeted therapeutic strategies and the advancement of medical knowledge in the field of heme biology.